Hello everyone. Today we are here to talk on a very interesting topic, which is anatomy and physiology of the vestibular system. I am Dr. Rahul K. Jaiswal, Associate Professor at the Department of ENT. So before we go into the topic per se, we must understand that the inner ear is a very interesting area. It has got a bony part and a membranous part. The canals that you see in this picture are three canals: anterior canal, lateral canal and the posterior canal. They open up in the ampulla, and then there is the vestibular nerve, the cochlear nerve and the cochlea. So the inner ear transforms fluid waves to electrical waves. So whatever we are talking is converted into like a transducer in the cochlea, and then it will be taken up by the nerve and sent to the brain. Next is, what are the inner ear is composed of? The inner ear is composed of bony labyrinth and membranous labyrinth. The bony labyrinth has got three parts. One is the cochlea, other is the vestibule, third is the semicircular canal. So there are three semicircular canals. One is the anterior, other is the lateral, and third is the posterior. Membranous labyrinth, which is found inside the bony labyrinth, are organ of Corti, that is in the cochlea. This is for sensory organ for hearing. Utricle and saccule in the vestibule, these are sensitive to linear acceleration. And semicircular ducts in the semicircular canal, they are responsible for angular acceleration. So the vestibular system, it lies in the otic capsule in the petrous portion of the temporal bone and has got five distinct end organs. Three semicircular canals that are sensitive to angular accelerations of the head rotations. Otolithic organs, that is the utricle and the saccule, that are sensitive to linear acceleration. So this is how, whenever we are in motion, how we maintain our balance because of these end organs. So the vestibular fluids. The vestibular fluids are perilymph and endolymph. The perilymph is present in the bony labyrinth. It is similar to extracellular fluid. It has got low potassium and high sodium. The endolymph, it lies in it's present in the membranous labyrinth. It is similar to intracellular fluid, high potassium, low sodium, and it is continuous with the endolymph of the cochlea. It is secreted by epithelial cells continuously and drains from the inner ear into the venous sinuses in the dura mater of the brain. So this is how the vestibular fluids are distributed. Next we'll come to the otolithic organ, utricle and saccule. From this picture, if you can see, this is the saccule and this is the utricle. So the otolithic organ, the sensory organs of the utricle and the saccule are the macula. Each macula consists of hair cells and supporting cells. The ciliary bundles of the hair cells project into the overlying gelatinous matrix known as the otolithic membrane. And otoliths are minerals and protein particles embedded in the otolithic membrane, that is the calcium carbonate crystals. So this picture, as you can see, this is the gelatinous material. These are the calcium carbonate crystals. This is the macula, and here the hair cells are there, and these are the stereocilium and the kinocilium. So this is how the otolithic organ, utricle and saccule are having the sensory organs, the macula. So the macula, it consists of supporting cells and hair cells. It detects and responds to position of the head in respect to linear acceleration and pull of the gravity. So each macula consists of thousands of hair cells that synapse with the sensory nerve ending of the vestibular nerve. And each hair cell has a 60 to 80 small cilia called stereocilia plus one large cilium called kinocilium. The kinocilium is always located in one side, and the stereocilia gradually become shorter. Minute filaments connect the tip of each stereocilium to the next longer cilia and finally to the kinocilium. So if you can see from this picture, this is what we've described. These are the small stereocilia, the larger one is the kinocilia, and these are the otoconial cells. The calcium carbonate crystals are above this. So this is how this is there, and these are the supporting cells of the macula. So the macula, when stereocilia bends in the direction of the kinocilium, it opens up potassium channels at the tip of the stereocilia, allowing potassium ions to enter and depolarize the cell. In response to depolarization, calcium gated channels are also opened up and calcium influx happens and leads to formation of action potential. So this is how it is happening. The potassium is entering after the stereocilium is moving. Once it is opening, the depolarization happens, the calcium channel is opening, and calcium channel enters into the nucleus, into the cell, and as a result action potential is generated. Now conversely, bending of the stereocilia in the opposite direction backward to the kinesilia reduces the tension on the attachments, and this closes the ion channels, causing receptor hyperpolarization and inhibition of the cell. So this is how the hyperpolarization occurs. So when the head is upright, the hair projects upwards into the gelatinous material. When the head is bent forward, backward, or to the sides, the hair cells are stimulated as the gelatinous material of the macula sag in response to gravity, causing the hair cells to bend. Stimulated hair cells signal nerve fiber, resulting in impulses traveling to the CNS on the vestibular nerve of the vestibulocochlear branch of the vestibular nerve and informing the brain that the head is in new position. Brain responds by sending motor impulses to the skeletal muscles in contract or relax to maintain the balance. So this is the utricle. You can see this is the utricle. The macula lies in the horizontal plane of the inferior surface of the utricle, so it determines orientation of the head in upright position, senses motion in the horizontal plane, that is forward movement, backward movement, left and right movement or combination. The saccula in the macula lies in the vertical plane and senses motion in the sagittal plane, that is up movement or down movement. Okay. So this is how this picture shows the macula, and this is the head bending forward. When the head is bending forward, the hair cells get stimulated and gravitational force pulls it downwards. So this is how the hair cells are working in macula and the utricle, saccule of the utricle and the saccule. The semicircular canals. The semicircular canals are three. One is the lateral or the horizontal, other is the anterior or superior, and third is the posterior. The semicircular canals are connected to the utricle at their bases. They are at right angles to each other. At the end of each canal, there is a chamber which is called as the ampulla, and the ampulla contains sensory receptor which is called crista ampullaris. So this picture shows the crista ampullaris, the sensory organ of the semicircular duct. Now the crista contains gelatinous material called the cupula. Embedded in the cupula is the hair cells. The basal membranes of the hair cell synapse on the sensory nerve of the neuron of the vestibular nerve. So the crista ampullaris detect and respond to angular acceleration and deceleration of the head. Rapid turns of the head or body stimulates the hair cells of the crista ampullaris. Appropriate rotation of the head in one direction bends cilia in the opposite direction, leading to depolarization of the cell. And the nerve fiber sends impulses to the brain and the cerebellum. Analysis of information allows the brain to predict the consequence of the rapid body movements and signal appropriate skeletal muscles to maintain balance. So this picture shows that if the rotation is to the left, the ampulla is moving to the right. If the rotation is to the right, the ampulla is moving to the left. So each semicircular canal works in concert with a partner located on the other side of the head, which has its hair cell aligned oppositely. There are three such pairs. Two pairs of horizontal canals, one superior canal each working on each side, and the posterior canal on the other side. The head rotation deforms the cupula in opposite direction of the two partners, resulting in opposite changes in the firing rates. For example, the orientation of the horizontal canal makes them selectively sensitive to rotation in the horizontal plane. More specifically, the hair cells in the canal towards when the head is turning are depolarized, while those on the other side are hyperpolarized. For example, when the head turns to the left, if you can see from this picture, if the head turns from the left, the cupula is pushed towards the kinesium in the left in the horizontal canal. The firing rate of the relevant axons in the left vestibular nerve increases. In contrast, the cupula in the right horizontal canal is pushed away from the kinesium with a concomitant decrease in the firing rate of the related neurons. If the head movement is in the right, the result is just the opposite. So, thank you for your patience. This is the anatomy and physiology of the vestibular organ, and in order to maintain this kind of balance, the vestibular system needs to be very corrected. Thank you.